Okay, we'll get started with the review first. So we'll start with chapter 6, and we're going to go through all the way till chapter 10. Okay, so you should be relatively familiar with respiration photosynthesis by now. You should be very familiar with this equation, okay? You can go forward and backward, right? If you start with glucose, you typically are going to break down glucose to form your ATPs. That's cellular respiration. You can go in reverse. You have energy. Instead of ATP, you're using light, and you're using CO2 and water in order to make glucose, and then the byproduct is oxygen. That's photosynthesis, okay? You should definitely know the processes. You should definitely understand the difference between NADH and NADPH, okay? Right? This is an electron carrier that is used to dump electrons off into the electron transport chain, right? This is made to be converted back into NAD, right? NAD is important for the glycolytic pathway, okay? NADPH is very different than NAD. We'll talk about that in a minute when we go to chapter 7, okay? You should know the cycles and the different metabolic pathways that are incorporated in the cell, right? In glycolysis, it occurs in the cytoplasm. Citric acid cycle in the ETC is inside of the mitochondria, okay, that organelle, right? These are separate from one another. Glycolysis can occur or perform without oxygen, okay? Right, glycolysis can work without oxygen. It will then go into the fermentative pathway, right? Glycolysis in comparison to respiration, if you just have glycolysis, it's very inefficient, okay? Glycolysis only gets a net gain of 2 ATP. The only way you can get your ATPs is if you remove that NAD, or you remove the electrons with NAD, okay? If you don't have this step, right, then you don't have your ATPs, okay? Remember, one, glucose gets broken down into two separate 3-carbon molecules, right? But in the beginning, you need to add 2 ATP. So one phosphorylation event to either side of that glucose molecule, right? You have 6 carbons. If you add the phosphates to the end, it makes it unstable and they break apart, right? And this is where you get the 2 separate 3-carbon sugars that can plug into this pathway, right? NAD needs to come in. Again, I'm stressing this so much. NAD needs to come in and remove the electrons in order to gain your phosphates for those ATPs, okay? You do not have this step 2 shown here. You do not have ATP. You do not have energy, okay? This is why fermentation is important, right? Because if oxygen is gone, that means the citric acid cycle is not working, right? The citric acid cycle requires oxygen, okay? Right, we talked about the citric acid cycle here. You require, well, I should rephrase that. You require the ETC to work. And the electron transport chain picks up the electrons from these NADHs and these FADHs in the Krebs cycle, as well as in the glycolytic pathway, okay? These need to dump off and be reused, okay, to go back into NAD. And in order for them to lose their electrons, they have to come to the electron transport chain and dump their electrons off. This requires oxygen, okay? See this oxygen molecule here? In order for the electron transport chain to work, you need to have oxygen. In order to dump NADs, or to dump NADH's electrons off, you need to have oxygen, okay? Oxygen is important. If you don't have this, you don't have the working electron transport chain, the mitochondria does not function. You cannot dump the electrons off, okay? This is for humans, this is for us too. You can have anaerobic respiration as well, but they use different species, so like nitrates and nitrites, okay? But again, you need oxygen for this to work, right? If you don't have oxygen, this electron transport chain doesn't work. The hydrogens do not pump, okay? But this reducing power shown here, this NADH, these NADHs, and these FADs, they need to be recycled. If the electron transport chain does not work, right, the cell must perform something called fermentation, okay? humans perform lactic acid fermentation. And what happens is fermentation, the whole sole purpose of fermentation is under anaerobic conditions. When you do not have oxygen, that's fueling, or that's allowing for this electron transport chain to function and work, then you lack the ability to dump off these electrons from NAD, okay? This is where you get lactic acid fermentation, or you get alcohol fermentation in yeast, right? Yeast can perform both. They're just like us. They can either do respiration or they can do fermentation, right? We like them to do fermentation because it makes ethyl alcohol for us, but that is a toxin. Ethyl alcohol is actually not good for the cell. It's not beneficial at all, okay? It's a waste product, right? That's why we can use ethanol as a sterilite or an antiseptic, okay? 70% ethanol kills almost everything, okay, right? So the whole purpose of fermentation, which I'm stressing, the whole purpose of fermentation is probably on your exam, is to recycle these NADs back, because there's no oxygen and the ETC is not working, okay? This is not working here. The electron transport chain is not working, so this cannot work. So the mitochondria essentially shuts down. You go through the glycolytic pathway. Instead of these NADHs coming back and dropping on the electron transport chain, they're going to be dropping on the organic molecule. So in fermentation, this is ethyl alcohol, right? This is acetyl acetate. This dumps off on the 2-carbon molecule here, and it makes ethyl alcohol, okay? CO2 is released as well, right? The whole point of this is to dump the electrons off for it to go back and be reused in the glycolytic pathway. And again, this is important because in the glycolytic pathway, you need the NADs to come back and remove the electrons in order for the cell to gain the ATPs, right? To perform work. Let me just preface it this way too. If you don't have any oxygen, the citric acid cycle, and this cycle is not working in the mitochondria, okay? Because look at how wasteful this would be. If you're not dumping, well, let me point out the main purpose of the Krebs cycle. The main purpose of the Krebs cycle is to get to NADs and NADHs and FADHs, okay? To get electrons. The whole purpose of this cycle is to gain electrons for the electron transport chain, okay? This will not function without the electron transport chain. So this does not function without oxygen, okay? Why? Well, think about it. You only gain 2 ATPs out of this cycle, out of 2 cycles. So 1 whole glucose only gives you 2 ATPs. It's the same as glucose. It's the same as glycolysis. But look at all of the waste products. You have 4 CO2s. You also have 2 FADH2s and 6 NADHs, okay? If the electron transport chain is not working, you need to make a whole bunch of alcohol or a whole bunch of lactic acid, okay, to dump these electrons off, okay? Because you need NAD to go back into the glycolytic pathway. This is wasteful if you don't have oxygen, okay? Right? It's like burning gas with the car moving. You're wasting these molecules here. These molecules are important in glycolysis, and you're gaining the same amount of ATP for 6 reducing power, or I guess 8 reducing power molecules, okay? 8 electron carrier molecules, right? You don't need this to function if oxygen is not present, okay? So in humans, we perform lactic acid fermentation. In yeast, they perform ethyl alcohol fermentation, okay? So yeast, there's also different other pathways. There's succinate, there's fumarate's one. There's different types of fermentation, okay? But in humans, lactic acid, and yeast, ethyl alcohol, okay? All right, that's it for that, okay? So for photosynthesis, what you need to know, this was a little shorter, right? You need to know photosynthesis is very important for life on Earth, okay? Right, bam. You need to know about the pigments. We have photosystem 1 and 2, or first photosystem and second photosystem. You also have different types of pigments, right? So chlorophyll absorbs visible light, or in that spectrum is a very So again, visible light, the electromagnetic spectrum is a very small portion of the spectrum, right? You have between 380 and 750 nanometers, okay? Chlorophyll does not absorb green light. It's like blue, violet, and then orange and red. Okay, and there's two reactions here. And I just want to emphasize this as well. Okay, this is a reverse of respiration, right? Respiration goes from this, right? You need oxygen and glucose to CO2 and water. Whereas photosynthesis does a reverse. It's CO2 and water, okay? You have the light reactions and you have the Calvin cycle, okay? Very important that you understand what the light reactions are for, right? The light reactions use visible light in order to gain ATP as well as reducing power. So remember we talked about NADH, right? That needs to be recycled back into NAD for glycolytic pathway. This is NADPH. This can be stored in the cell. You want to make this in order to work the Calvin cycle. This is reducing power. This is different than NADH. Excuse me, I gotta sneeze. Oh, excuse me. So this is different than NADH, right? This is going to be reducing power. These electrons are used to build carbon molecules, okay? These electrons here are different than, this molecule here is different than than NADH, okay? Very different. Well, not very different, but different, okay? NADPH is used to build carbon molecules, okay? You need that for carbon fixation, right? CO2 from the atmosphere gets brought in so you can make glucose, okay? You need 6 CO2 molecules to make glucose. Also, ATP can be harvested from the light reactions, okay? Let's go to here, photosystem. The photosystems work by the light being excited by a photon. Then you can actually increase that electron to a higher energy state. So the excited energy state, and that can be used for energy, that can be used for carbon fixation, right? You're passing off those electrons to the NADPH, all right? Okay, so this occurs in the photosystem, the thylakoid membrane. Read this, go through that. Okay, so again, you have the first photosystem, right? You remove the electrons from water, so you require water in the light reactions of plants, right? So water comes in, you remove those electrons, you gain those electrons from water, they get excited to a higher energy state. You have an electron transport chain. This is used to make ATP, okay? This is important because this powers the dark reactions, okay? This ATP is not, well, I guess I'm giving you an answer on the exam, but I don't care. If you watch the review, you watch the review. ATP for this is going to be utilized for the dark reactions, but plants also have mitochondria, okay? Plants have both chloroplast and mitochondria. And you're like, why? They can make ATP, they can store glucose, right? This ATP isn't enough. This ATP is utilized in the chloroplast, and it's utilized for the dark reactions, okay? And again, those dark and light reactions can work separately. But this ATP from photosystem 1, or the first photosystem, again, is used for the dark reactions. It's used to make glucose. This is work. This is energy to perform work in the form of fixing those carbon molecules together to make glucose, okay? Second photosystem, again, the electrons get dumped off. It gets excited again. These electrons are then passed on to NADP, and then to make NADPH. Again, this is going to be the electrons in the form of reducing power, okay? So this will be used, these electrons will be used to dump on the carbon molecules to make glucose, okay? All right, so here you go. That first photosystem, again, it's the electron transport chain. You have different embedded enzymes in the membrane that are gonna be pumping hydrogen into this space here, right? So the inside the thylakoid membrane is acidic, okay? This acidity is going to be, or this electrical chemical gradient, right? We talked about this. This is also important in respiration as well in the mitochondria, right? The acidified environment here is going to be used. These protons are going to be used to pump through the ATP synthase complex, right? This makes ATP. This goes to the dark reactions or the Calvin cycle, right? So electrons to build the carbon molecules and the energy for the work that's required to build the carbon molecules, okay? Again, ATP synthase. Plants also have mitochondria, right? Because they actually need ATP for other cellular functions. This is going to be kept inside of the chloroplast. And photosystem 1, again, ATP. Photosystem 2, reducing power in the form of NADPH in the carbon, and it's used in the Calvin cycle, okay? So again, this is very similar to how the mitochondria works, right? Very, very similar. You have an acidified internal membrane in the thylakoid, or acidified space. This is an electrical chemical gradient. Remember, it's a dam. The dam is holding back the water. Think of the water as these protons, right? Electrical chemical gradient. This enzyme, ATP synthase, allows for the passage of a proton or protons through to form ATP, okay? All right, so the Calvin cycle is going to be utilizing the light reaction products to make sugar, okay? So it pulls in CO2 and to make sugar, okay? Do you remember what comes in and comes out of these different cycles, right? Water and light are used, and then you get oxygen released by those plants or photosynthetic organisms, and then you have CO2 coming in and sugar coming out, right? CO2 gets carbon fixated with these two, with ATP for work and NADPH for the electron sources, okay? These can happen independently, okay? So that's another thing that you do need to know. So there are, so the stoma is what's important here, okay? The stoma will only open if the cell or the plant has enough water in order to perform the different tasks, okay? The stoma needs to open to allow CO2 from the atmosphere to come into that plant, though, into those chloroplasts, though, okay? The stoma will not open in certain plants until it's night, okay? So this is going to be cactus or plants that live in the desert, right? These plants cannot open their stoma because guess what? At 115 degrees Fahrenheit in the middle of the day, you will dehydrate really fast, okay? So opening the stoma to let CO2 in is not favorable in those environments. So they'll wait till night. So these separate, the light reaction, the Calvin cycle can be separated, right? The organism or the plant can store, or this photosynthetic organism can store this NADPH until they open the stoma and allow CO2 to come in order to perform the Calvin cycle, okay? So these are two separate reactions, okay? They're in the same organelle, but again, certain plants live in very, very harsh conditions, so it's important to be able to store the ATP and the NADPH until a favorable condition occurs, and they can actually perform this, okay? Other plants do not care whatsoever, right? Plants that have plenty of water, in very nice environments, very temperate environments, or even in the Amazon, right? These can be occurring simultaneously and very, very rapidly. Those plants can grow very, very fast, like bamboo or other plants in the Amazon. They grow exponentially, right? Because you need enough nutrients, they need enough water, they got plenty of sun, and they grow, okay? Whereas cactus or cacti grow very, very slowly for this reason, right? They need to separate out these reactions because at night, they can perform the Calvin cycle, but during the day, they'll perform the light reactions, okay? All right, and the Calvin cycle, essentially what you're doing is pulling in CO2 from the air and fixating it. So NADPH comes in, dumps the electrons off, cool, right? You need ATP from the photosystem 1, and then you also have rubisco. And I know we talked a little bit about rubisco. The most abundant and probably one of the most important enzymes in the world, right? Rubisco is able to grab CO2 from the air and fix it to make a sugar molecule glucose, okay? But yeah, don't worry about all these separate steps. Just know that the Calvin cycle is where you dump the NADPH and the ATPs are used in order to make your sugar, okay? go over this slide, right? This is important here too. All right, time's up. So cellular reproduction, let me see. All right. Know these: cell replacement, cell growth and division, and asexual reproduction, mitosis, okay? Know the differences between mitosis and meiosis. Mitosis is to make somatic cells, or to make cells that are normal cells in the body, like your skin, tissue, organ cells, things like that. Meiosis, right? Meiosis is a little bit different. Is going to be used to make sex cells, okay? So, yeah, gametes are made through sex cells. So you have different proteins associated, or different structures associated with the eukaryotic chromosomes, right? We have histones that wrap around DNA. You have chromatin. This is bundles of DNA, the protein and DNA combined, and the nucleosome. These are going to be bead-like units of DNA packed together, and they're going to be tightly wound, right? Now, there's a varying number of chromosomes in different organisms, right? Not all organisms have 23 chromosomes like we do, or 23 homologous pairs, 46 different chromatids, right? DNA packing. So this is how a chromosome is. These are histones. These are the nucleosomes, which are the bead-like structures, and you have the DNA tightly wound in the chromosome, okay? So we do have something called sister chromatids. Yeah, they're homologous pairs. So homologous pairs, remember, you have 23 pairs, one from your mom, one from your dad, okay? And it's important that you note the differences, and you're able to describe that, right? One from your mom, one from your dad. They are different, but they contain the same genes, right? They're different, right? It's like two people come together. We're both people. We both have, similar genetics, but we have differences in each other, okay? It's the same thing as what homologous pairs are, okay? So you need to be able to differentiate that, okay? Sister chromatids are a little bit different. They're just duplicates of each other, right? And this is going to be done during mitosis or meiosis, depending on the stage. Cell cycle, this prevents against cancer, right? There's certain steps that the cell needs to take in order to perform interphase, telophase, cytokinesis, all of that, okay? These are the different stages. Interphase, the majority of the time is spent here. You have prophase, right? The forming of the centromere, the breaking apart, the joining together of the DNA and condensing of the DNA. Metaphase is when it lines up. Anaphase, separation. Telophase and cytokinesis, right? Telophase is the breaking or the invagination of the membrane, and then cytokinesis is the actual separation of that, okay? You can go through these steps. It's good to go through them. There's a cleavage furrow, again, the invagination of that membrane in order to split into two separate daughter cells, okay? The difference between plants and mammals is that there is a cell plate, okay? The cell plate needs to form because the cell wall, cellulose, is very rigid, very sturdy, and it provides an issue for the separation of those, okay? Those cells during cytokinesis. Cancer cells, go over this, right? We talked about cancer and what cancer is. Yeah, and remember, it's abnormal cell growth, okay? Tumor, radiation therapy, meiosis. So we talked about meiosis, sex cell formation, right? It's going to be a little bit different than mitosis, and we do have comparisons, right? You should know the differences between somatic cell, karyotype, homologous chromosome, right? Go through what an autosome is, right? You should know diploid and haploid, right? Diploid, meaning 2N, is your somatic cells, and the somatic cells, remember, are going to be any cell found in your body that's not a sperm or an egg, okay? And these are going to be diploid. What does that mean? Well, they have both homologous pairs, right? You have one from your mom, one from your dad. If you're haploid, you have half the amount of the chromosomes that you need, right? You have only one from your mom or one from your dad, and these are your sex cells, okay? That's 1N, okay? This means that your sex cells, your gametes, your eggs, the sperm, is only going to have one copy, either from your parent, either from each parent, right? And remember, during metaphase 1, you have something called crossing over, okay? Metaphase 1, I believe. Crossing over, okay? So this is also going to further mix up and cause genetic variation, okay? So again, diploid, these are somatic cells. These are all your cells except your gametes, right? The gametes are going to be haploid, okay? Sorry, prophase 1 is when you have crossing over. Metaphase is different. So prophase 1 is crossing over event, right? So prophase 1 is when the homologous pairs will come together and start exchanging genetic information, right? So remember, those homologous pairs or homologous chromosomes are going to be the ones that come one from your parent, one from each parent, okay? And they can exchange DNA to further mix up genetic variation in your sex cells, right? So that is one form of genetic variation. The other part of genetic variation that you see is going to be in meiosis 2 during independent assortment, right? So each of these is going to break apart, and then you're going to have your haploid cells. So the separation of those two homologous pairs are going to cause a variation event as well, right? You can have chromosomes mixed with your moms and your dads, and then vice versa, especially in prophase 1. You'll have that crossing over event that'll cause that increase in variation as well. And again, meiosis is just to create variation, right? Just to create an increase in variation in your sex cells. All right. We talked a little about non-disjunction or issues with the separation. You could have Down syndrome. You have different types of genetic disorders if you have extra or you're missing a chromosome as well. Okay. We talked a little bit about this. Yeah, that's it. Let's see. 9 patterns of inheritance. So I will ask you to understand the Mendelian Punnett squares. I'm not going to have you do a dihybrid cross on the exam. That's just going to take too long. So I may ask you to do a dihybrid cross later, maybe in the next exam, because I kind of want you to do one. But you will have to perform or understand how to perform a single cross, okay? All right. So understand the statistics behind it. You should be able to define genotype and phenotype, and you need to understand genotype and phenotype, right? Genotype is going to be the actual genes, right? So like you can be heterozygote or you can be homozygote, whereas phenotype is going to be what's expressed, right? So these yellow seeds all have that same phenotype, but they have very different genotypes, right? So the yellow seeds are going to have heterozygote or homozygote dominant, okay, right? So homozygote, so 2 R's is going to give you yellow, but a seed as well, okay? Or, I'm sorry, it's the Y's types, okay? That's